Yes, good afternoon to all. Today we are going to discuss the summary of the chapter Strategies for Enhancement in Food Production. Then see some of the questions at the end of the chapter as the third assignment. So we can, first of all, we can see the summary of the total chapter. What are the strategies for enhancing the food production in India? Totally we divide the chapter into two categories, two parts. First comes the animal breeding and then comes the plant breeding. We divide the total chapter into two, animal breeding and plant breeding. And animal breeding is otherwise called as animal husbandry. And this animal husbandry or animal breeding include the breeding and growing of plant animals which are for human use, that is called as animal breeding. And the aims of animal breeding, we are trying to improve the quality of animal products. We are trying to improve the quantity of animal products, and we are using to try to combine the characters of two animals, two breeds together by crossbreeding. So these are the aims of animal breeding. And we have seen that animal breeding, four examples are given. One comes the dairy farm management, that is already discussed. The growing animals which are related to milk and milk products is called as dairy farm management. And when we are starting a dairy farm, when we are growing cow, we have to look after some important things to be considered while we are carrying that cow, or we are starting a dairy farm. And we discussed that we have to provide best shelter, we have to select the best type of breed, and we have to give medical checkup, we have to provide good type of food to them. So these are the things to be cared when we are starting a dairy farm. Then see, second comes the poultry farm management. How we can start a poultry farm, that is related to birds, related to that, breeding of birds and like animals are called as poultry farm management. And we have to care, we have to select the best breed for that, and we have to provide food, and we have to give electricity or power supply or heat for incubation and hatching of the egg. And we have to care one disease about that, H1N1 disease, virus, bird flu virus comes to that case. And related to third one comes the fisheries, comes the apiculture, we call as honeybee, growing of honeybee is called as apiculture. The common species of honeybee grown in India are called as Apis indica. And economically and biologically, apiculture is very important. Biologically, they are providing pollination and increasing the productivity of cultivating plants. Economically, we are getting honey, and wax are the products of apiculture. Honey is used for getting medicine and used as food, and the wax is used for making candle or cosmetics or polishes. So that is already discussed. Then comes the fourth, and you see that honeybees are a very social group of insects. They have three types of individuals in a colony. That also we discussed. Then comes the fourth example is given, fisheries or pisciculture, and growing fishes and aquatic organisms like crab or prawn, fishes, etc. are called as pisciculture. Then comes they are economically important because they are controlling the economy of a country, and further we are getting various products we are getting from these organisms like oil or medicine or base for manufacturing of the fertilizers. Like that we are getting various products from that by fisheries. So 4 points we discussed. Then comes the methods of animal breeding. We classified into inbreeding and outbreeding. Inbreeding, outbreeding. Inbreeding means breed means a group of similar organisms having a common ancestry is called as breed. And crossing between two related organisms of a breed, very closely related organisms are called inbreeding. Crossing of very closely related organisms is called as inbreeding. As a result of inbreeding, we are getting a process called phenomena called as inbreeding depression. That is the loss of superior character due to continuous inbreeding is called inbreeding depression. So good character will lose generation after generation, that is called as inbreeding depression. So, and to overcome that we are doing the process of outbreeding, and there are three methods of outbreeding. Outbreeding means crossing between two different organisms of the breed are called outbreeding, or crossing between different organisms, unrelated organism is called as outbreeding. And there are three methods, one called as outcrossing, second is called crossbreeding, and third called as interspecific hybridization. Three methods are there. Outcrossing is the crossbreeding and interspecific hybridization. And first comes the outcrossing. And outcrossing means crossing between organisms of same breed is called outcrossing, very closely related organisms of a breed. So inside a breed, inside a group, very closely related organisms, they are crossing, that is called as outbreeding. And crossing between two different breeds are called, two different breeds are crossed, that is called as crossbreeding. And example is given, Fishery is an example. Fishery is an example given for that. That is a type of sheep developed in Punjab, crossing Bikaneris and Merino rams are crossed together, so getting a sheep in that is developed in Punjab. Then comes the interspecific hybridization, and that is crossing between two different species. That is not possible in the nature. But we are crossing two different species, different types of unrelated organisms. We are crossing two species together, that is called as interspecific hybridization. The example is given, mule is an example, and which is sterile, it cannot reproduce, and mule is obtained by crossing male donkey and female horse is crossed. So we are getting a species called mule, and as an example for interspecific hybridization. Like that, crossing can occur between two species also. So there are three types of outbreeding, outcrossing, crossbreeding example, Fishery is important, interspecific hybridization. And then we discussed what is the method of animal breeding, and during the animal breeding we are doing artificial insemination is given. That is, male gametes are collected from male animal, semen is collected, male gametes are called as semen, collected from male animal and injected in the body of female, reproductive tract of female animal, that is called as artificial insemination is conducted for crossing. Similar to our cross-pollination, pollen grains are collected and deposited in the stigma of the female flower. Similarly, instead of pollen grains, we are collecting male gametes or semen is collected from male animal and injected into the body of female animal, that is called as artificial insemination is conducted. And for that purpose, we are storing the male gametes at the temperature of minus 196 degrees Celsius, that is the temperature of liquid nitrogen. Male gametes can be stored for, or semen can be stored for long time in semen bank. Institution where gametes or semen is collected is called as semen bank. So male gametes having a short viability lifespan, so they cannot be preserved for long time. So we are keeping the male gametes, male semen at a temperature of liquid nitrogen minus 196 degrees Celsius. Such a preservation of gametes at a low temperature is called as cryo preservation. That is a new term for you, cryo preservation. Gametes or any cell, pollen grains or whatever may be collected and can be stored at a low temperature called as cryo preservation. And so that is about the artificial insemination. Then comes the latest concept called MOET, multiple ovulation embryo transfer technology, MOET. That is the latest achievement in animal breeding, multiple ovulation embryo transfer technology. And during that process, we are by using hormones like female hormone, follicular stimulating hormone, FSH, we are producing numerous ova in a female animal instead of one ovum. that is called as super ovulation. Numerous ova are produced, and we are making artificial insemination. So the numerous ova fertilize into numerous zygote, and zygote start to develop. When the zygote began to divide into embryo, the developing embryos are collected, taken out of the body, and they are introduced into other female recipient organism called as surrogate mother. So the zygote is transferred to different female animals. They will grow in the female animals as young ones. So we can simultaneously, within a short time, we can produce numerous organisms, genetically similar organisms can be produced by multiple ovulation embryo transfer technology. And this is the types, these are the main methods of animal breeding. Then comes plant breeding. Plant breeding, the growing and the cultivation of plants which are used to human beings, called plant breeding. And plant breeding started along with the Green Revolution, or when plant breeding started. Actually, this is started with domestication. 10,000 years ago, when agriculture started, we began to cultivate the plants, and same plant is cultivated continuously, so it will result in the inbreeding depression. On the other side, human population increases very fastly, so we cannot satisfy the requirement. So after independence, 1960s, in India and from different parts of the world, they began to produce numerous varieties which are high yielding and disease resistant varieties are produced in different parts of the world. In India, and Green Revolution started in 1960s under the leadership of Dr. M. Swaminathan, leading agriculture scientist of India at that time and now also. And he started Green Revolution, and by introducing different types of varieties of rice and wheat from different parts of the world, of Japan or from Taiwan or from some other countries, rice or wheat varieties are introduced to India, and that is supposed to be the basis of Green Revolution. And by using the introduced varieties from different parts of the world, we produce new varieties here by crossing with our local variety. Introduced plants have some good character. Our native plants have some other good character. So we combine the characters by the process of crossing or cross hybridization, and we discussed what are the methods of producing new varieties in India. First of all, we discussed 5 points are there. First of all, collection of variability from different parts of the world. We are collecting the varieties, different varieties of the plant. Second point, we test and select 2 plants. So studying, and we are selecting. So by analyzing the character, we select 2 plants. 2 plants were called as parent plants, and these parent plants were cross-breeding, hybridized together. They are crossing together, and pollen grains are collected and deposited in the stigma. That is called as cross-pollination. During that, we are doing 2 things. Remember that: emasculation and bagging. You remember the 2 points: emasculation, bagging. Removal of anther from bisexual flower is called as emasculation, and it is covered by a polythene bag. Emasculation is to prevent the to prevent the self-pollination. We are doing emasculation, and bagging is done to prevent the unwanted pollen grains. So we are doing emasculation, and then cross hybridization, again covered with the bag, and pollen grain seeds are collected and seeds are produced. That generation is called F1 generation. And then after that, we are testing and selection of superior recombinant. From that, thousands of plants are produced. From that, we select the plants with good character, and they are tested in various parts of the country. Then from that, we select the superior recombinant. Then they are tested for its performance. Then it is released, and after releasing, it is commercial. So testing, releasing, and commercialization. 5 points are there. Production of the variety. What are the steps of creation of a variety, and then how to get that 2.5 marks for that. 5 points are there. Then plant breeding. Then for what purpose we are using plant breeding? First of all, we selected. We are breeding using the plant breeding for production of disease-resistant variety. We are producing disease-resistant varieties in India for combining for avoiding the diseases. Then comes plant breeding for insect resistancy. insect resistance can be produced by the method of cross breeding. When we are doing disease resistant variety, we have a crossing method or maybe mutation method is there. Sudden heritable changes are called mutation. We can cause changes by mutation also. Then we are discussed for improved food quality. We have to improve the food quality. We have to improve. At that time, there comes a process called biofortification. We are growing and the cultivation of plants with a high level of nutrient is called as biofortification, to improve the oil content and quality, vitamin content and quality, micronutrients and like that minerals. Then comes the disease resistance. Then comes the another method called important concept called single cell protein. Microorganisms or bacteria or fungus or cyanobacteria can be used as a source of protein, that is called a single cell protein. Methylophilus methylotropous bacteria, it will produce 250 grams of Methylophilus methylotropous can produce 25 tons of protein during its life cycle. So called as single cell protein is an important concept. We can satisfy the requirement of protein requirement to whole living organ or human beings by the concept of single cell protein. We can grow the bacteria, cyanobacteria in wastewater so that we can purify the water, or we can use it water treatment, biological treatment you studied in the third chapter swallowing sometime. So the single cell protein bacteria can be grown in wastewater, so we can use the purification of wastewater can be done. And on the other hand, we can grow these microorganisms, can be made into capsules and can be eaten to satisfy the requirement of protein content. So these are all the concept of, and of that biofortification and single cell protein is very important from plant breeding. And here remember the cross breeding, interspecific hybridization, MOET and artificial insemination is very important, that comes from the animal breeding. And last we discussed the latest achievement of plant breeding called as tissue culture. Growing tissues or plants in nutrient medium is called tissue culture. And we are selecting a plant part, cell or leaf from the parent plant. That plant is called explant. We are keeping the explant in nutrient medium under controlled conditions by providing hormones. And if control all the conditions in the room or laboratory, the cell will, the plant explant will divide and form a mass of tissue called as callus. And callus, to that callus we are adding proper amount of cytokinin and auxin hormones like 2,4-D or benzyl aminopurine. On the callus organs will develop. When we are adding auxin, roots will develop. And when we are adding cytokinin, shoot bud will develop in the callus. Formation of root and shoot bud is called as organogenesis, organ formation. Then the plants are separated and the small plantlet grown in the test tube. Then we are going to the field to see the send the unfavorable environmental condition. They have to face that. So after their plants are made in the laboratory, they are grown in the outer medium or on the field. So that is about the tissue culture method. And when we are using, what are the applications of tissue culture are more important than the procedure of tissue culture. Applications are continuously asking for examination. And micropropagation, somatonal variation, more than that, production of virus-free plants can be produced by meristem culture. And last comes the somatic hybridization of combining protoplasm technology, combining protoplasm of two species together. Example is given as tomato, which is obtained by crossing potato and tomato. So these conditions are also. By this we complete the chapter and revise. Again, revision is given to understand what is the any difficulties there. Then see the question is given to you. Question is already, the exercise questions are there. About questions are given to you. Already carries 12 questions are there. The questions are already sent by you yesterday itself. So we can discuss, and you write at the third assignment on today's date, 27th July. You can write the assignment as third one. I hope that you completed the two assignments given already given the previously I given that. So I will discuss the questions and you can write the answer very clearly. First question: explain in brief the role of animal husbandry in human welfare. What is the role of animal husbandry in human welfare? Animal breeding. And animal breeding, first of all, you write what is animal breeding. Growing and caring of animals which are useful to human beings are called as animal breeding. And main aims are, you write 3 or 4 aims: to increase the yield of animals, to improve the desirable quality, we have to improve the quality like milk production or like that. Then to combine the good characters of 2 breeds together. See, you write 3 points: to increase the yield of animals. Second point, to improve the desirable quality. Third point, to combine the good qualities of 2 breeds together by cross breeding or cross hybridization. You have 3 points are there and you have very clear. Then the second question comes: if your family owned a dairy farm, what measures would you undertake to improve the quality and quantity of milk production? If you are developed or if you are owning a dairy farm, what things you can take to improve the quality? So first of all, the method is called dairy farm management, and quality can be improved by what methods we can adopt for that. First of all, we can select good breed for starting the dairy farm. Selection of breed should be, the selected breed should be high yielding and with good quality. Second point, we can provide best shelter for them. Third one, we have to give nutritious food material. Fourth one, we have to provide regular medical checkup should be provided. Then fifth one, we have to provide clean, healthy and hygienic conditions by providing adequate water for cow shed. So remember, these things will be given in the notebook. You write the same point. What are the methods to improve the quality of milk production? Third one, what is meant by the term breed? What are the objectives of animal breeding? Again the question comes, what is a breed? Breed means a group of similar organisms, genetically similar organisms with the common ancestry is called as breed. See, in plants we are using the term variety. Breed. Animals we use the term breed. B-R-E-E-D. Breed means a group of genetically similar organisms having a common ancestry is called as breed. It is the subdivision of species can be again divided. So that comes the smallest group called as breed. Group of genetically similar organisms having a common ancestry called as breed. And what are the objectives of animal breeding? You already discussed that. To increase the yield, to improve the desired quality, they combine the good qualities of 2 breeds together. Then fourth one, name the methods employed in animal breeding. According to you, which of the method is best and why? This is important one. Questions are repeatedly asking. Name the methods of animal breeding. Methods of animal breeding: inbreeding and outbreeding. According to you, which of the method is best? Inbreeding is best one or outbreeding is best one? According to you, outbreeding is the best method. Why? So answer is that first part, inbreeding and outbreeding. You define that inbreeding and outbreeding firstly. Then you write outbreeding is the best method because it will combine the different qualities together. By inbreeding result in inbreeding depression. So to overcome inbreeding depression, we are doing outbreeding. Outbreeding is the breeding of, crossing of unrelated organisms called outbreeding. It is the method of, which is the best method of animal breeding, and it will improve the qualities. It will help to combine the desirable qualities of 2 breeds together. So that outbreeding is the best method of animal breeding than inbreeding. You have to write the inbreeding depression. Because of, to prevent inbreeding depression, we are doing outbreeding. So you write like that also. Then comes the fourth question is very important. Fifth one, what is apiculture, how is it important for our lives. Growing of honeybee is called as apiculture. How it is important, second part, because it is important biologically and economically. It is important. Biologically, pollinators improve the productivity. Economically, wax and honey are the products and use the uses of honey and wax. Sixth one, discuss the role of fishery in enhancement in food production. What is the role of fishery? Fishery, growing of aquatic organism. It is economically important and it provides fertilizer, etc. It provides employment to a group of persons living in the coastal areas, because in most of our country share coastal areas. So it will decide the economy of coastal area. It will decide the life of coastal peoples. It is a type of culture. It provides employment. It provides foreign money. So like that, how it will affect the food production. Then briefly describe various steps involved in plant breeding. That is very important. Steps of plant breeding. 5 points are there. We will repeat once again. Select collection of variability, selection of parent, cross breeding, selection of superior recombinant, testing, releasing and commercialization. Important point. So you remember that, and you have to remember the emasculation and bagging. Then explain what is meant by biofortification. Growing of plants with a high level of nutrient is called biofortification. And what are the aims? Aims are also important. 4 points are there. Each carry half marks. You write the aim of biofortification also. Then comes which part of the plant is best suited for making virus-free plants and why. Usually meristems are used for producing virus-free plant, because meristems are usually virus-free. Apical meristem is usually without virus because of the absence of vascular tissues in the meristematic region, because virus is transported through xylem and phloem. Meristematic tissues are without xylem and phloem. So meristematic tissues are without virus. So they can be grown in the culture medium to produce virus-free plants. So answer is apical meristem is virus-free. So that is used for producing virus-free plant. Then 10th one, what is the major advantage of producing plants by micropropagation? So by micropropagation, what is the advantage of that? Micropropagation, we can produce thousands of genetically similar plants within a short time. Within a small period of time, we can produce thousands of genetically similar plants. They can call it as, we can use the term clones. They are genetically similar. So that is the advantage. We can produce thousands of plants within a short time. 11th question, find out various components of the medium used for propagation of an explant in vitro culture. When we are culturing tissues in nutrient medium, what are the components of nutrient medium? You can refer internet also. You can find out what are the usually we are using agar medium. We are using agar medium. In that we have to provide carbohydrates, we have to provide proteins, we have to provide vitamins, we have to provide micro and macronutrients, we have to provide. More than that, we have to provide hormones should be provided like 2,4-D, benzyl aminopurine, etc. should be also provided. So these are the main components of nutrient medium. You refer the internet or textbook and write the answer. The main components are there. 12th one, name any hybrid varieties of crop plants which are developed in India. You refer the textbook and find out 5 hybrid varieties are there. Column 9.1 is there, 9.2 is there. You can see different varieties are there, and whatever the case may be, last time questions are continuously repeating this 9.1 and 9.2 column. So whatever the case may be, you try to study the 9.1 column and 9.2 column very thoroughly. It is very important for examination. So the chapter is supposed to be completed, and we go to the next chapter called as very important chapters and toughest chapter of botany, second year, that is called as biotechnology. In the next class we can start that. That is very important. So you write the assignment and complete the notes. I already given the notes for you. So complete the notes and write the assignment very completely. And now also I am thinking about to conduct an examination, but I don't know how to conduct the examination in this method. Anyhow, we have to conduct an examination. I will discuss with your group leaders or teachers for that purpose. Then we can, so you write, complete the notes and complete the notes and the assignments within 2 or 3 days. And next class, I think we can start with the biotechnology. Okay, thank you.